We are a group of researchers from the University of Moratuwa's Computer Science and Engineering Department. Our team consists of Nuwin Abayanayaka, Ruin Dayananda, and Dakshitha Shuriarachi. Our project was supervised by Dr. Charith Chitraranjan. Before we dive into our research, let's understand the topic, identifying valence from FMRI data. But what exactly is valence, and what is FMRI data? FMRI stands for Functional Magnetic Resonance Imaging. It refers to a brain imaging technique that measures brain activity using changes in blood oxygen levels. It's a non-invasive method and has the highest resolution among various brain imaging techniques like EEG. Now, let's clarify what is valence. Valence refers to a measure of how pleasant or unpleasant our emotions are. It represents the core emotional experience that we encounter in various situations. On one end of the valence spectrum, we have positive valence, which represents feelings of pleasure, happiness, and joy. On the other end, we have negative valence, which represents emotions of displeasure, sadness, or distress. In this section, we'll explore the significance of predicting valence and why it holds great importance in our understanding of emotions. So, let's dive in. Predicting valence holds significant importance in the medical domain, particularly when it comes to mental health and neurological disorders. Predicting valence in human-computer interaction is essential for three reasons: user experience, emotional feedback, and emotionally intelligent assistance. Valence prediction plays a crucial role in legal decision-making, offering insights into the emotional context of legal cases. Now, let's delve into the current status of the domain of valence prediction and explore the exciting progress that has been made in recent years. The domain of valence prediction from fMRI data continues to be a subject of active research, with scientists tirelessly exploring new approaches to enhance accuracy and deepen our understanding. However, achieving sufficient accuracy for real-world applications in predicting valence from fMRI data remains a challenge. In 2015, a meta-analysis conducted by Lindquist concluded that there was no consistent neural pattern for predicting valence, although machine learning techniques were not extensively employed at that time. Since then, several studies have emerged, utilizing machine learning techniques to predict valence from fMRI data. These studies have made progress, but achieving high accuracies in valence prediction remains an ongoing endeavor. Let's explore what we did to gain insights into the underlying mechanisms of valence. Our research approach involved conducting a meta-analysis to explore the relationship between neural activation patterns and valence prediction. We also focused on developing a pre-processing pipeline and evaluating different machine learning methods, feature extraction and selection methods for accurate valence prediction from fMRI data. By integrating the pre-processing pipeline with a trained machine learning model, we created an integrated valence prediction model using fMRI data. Our meta-analysis examined the relationship between neural activation and valence prediction using a systematic approach. We screened a total of 2,317 papers using the Rayan AI collaboration platform. Meta-analysis was conducted using a generalized linear mixed model approach with a logit transformation. The evidence gathered from this process provides valuable insights into the neural mechanisms underlying valence prediction. The following is a high-level process diagram outlining the steps involved in model prediction. It encompasses 5 main stages: data acquisition, pre-processing and feature extraction, feature selection, prediction, and results interpretation. Let's explore how we obtain the necessary data for our research. Research in valence prediction faces the challenge of limited sample size due to the high cost of fMRI data collection, which requires specialized equipment, personnel, and expertise. Therefore, we have adapted datasets that were already collected from previous studies for our study. These are the datasets we have used in our study. Let's explore how we processed and extracted meaningful information from the raw fMRI data. In our research, we first utilized fMRI Prep, a standard pipeline for fMRI data pre-processing. The key steps involved were skull stripping, spatial normalization, slice time correction, T1 alignment, and head motion estimation. The following diagram illustrates the flow of the fMRI pre-processing pipeline. In the second step of our pre-processing pipeline, we utilized AVNI for further pre-processing. The key steps involved were head motion correction, smoothing, and scaling. The following diagram illustrates the flow and steps of the AVNI preprocessing. In the third preprocessing step, we applied convolution with the hemodynamic response function using AVNI. By convolving the FMRI signal with the HRF, we align the neural activity with the observed FMRI measurements, enabling us to accurately estimate the timing and magnitude of brain neural activation. Let's find out how we identify and choose relevant features that contribute to accurate valence prediction. We applied feature selection methods to identify voxels contributing to valence prediction: univariate t-test, principal component analysis, and independent component analysis. Univariate t-test yielded the best performance across all three datasets. The goal was to identify significant differences in voxel activation between positive and negative valence conditions. Let's explore the modeling techniques and classification techniques employed for our valence prediction. Our research utilized within-participant and across-participant classification. Within-participant classification determined valence by analyzing individual participants' brain volumes. We employed k-fold cross-validation for robustness and generalizability. Across-participant classification assessed similarities in valence representation across participants. We used leave one participant out cross-validation for evaluation. This approach revealed common patterns and individual differences in valence representation. Let's take a look at the models we applied. We employed several models, including support vector machine, Gaussian naive Bayes, logistic regression, random forest, light gradient boosting machine, artificial neural networks, and 3D convolutional neural networks. Note that for each model, we only tested for feature selection methods that were relevant and applicable to the specific characteristics of that model. In this section, we'll present the results of our valence prediction models and discuss the insights gained from our research. Here are the results we obtained for across-participant classification across our three datasets using three feature selection techniques: t-test, PCA, and ICA. It is evident that the t-test yields the best results compared to the other two techniques. Furthermore, based on the results, there appears to be no significant difference in performance among SVM, LR, ANN, and 3D-CNN. We achieved exceptional results for our third dataset, possibly due to the longer stimulus duration compared to the other datasets. However, it deviates from real-world usage compared to the other two datasets. Now, let's compare our best results with the previous ones. It is apparent that we were able to achieve significantly higher accuracies than previous studies across all datasets. This implies that our preprocessing techniques and feature selection methods are much more powerful than their predecessors. We present the outcomes of our within-participant classification analysis on three datasets, employing three feature selection techniques: t-test, PCA, and ICA. Furthermore, our findings suggest that the performance of SVM, LR, ANN, and 3D-CNN in this across-participant classification task exhibits no significant disparity. Within the scope of within-participant classification, the limited occurrence of events notably impacted the accuracy, resulting in lower performance when compared to across-participant classification. Now, let's compare our best results with the previous ones. It is quite clear that we were able to achieve much higher accuracies than previous studies across all datasets. This indicates that our preprocessing techniques and feature selection methods are far more powerful than the ones used in the past. Now, let's dive into the conclusions drawn from our research on valence prediction from FMRI data. The meta-analysis shows that the ability to predict valence from neural activation is significantly higher than chance level, indicating a meaningful relationship. Our study utilized a three-step preprocessing approach with FMRI prep and AVNI, including t-test feature selection, which greatly improved overall performance. SVM, LR, ANN, and 3D-CNN classifiers performed better than other algorithms. In conclusion, combining these classifiers with the preprocessing steps enables accurate classification of valence categories in fMRI images. Thank you all for watching and joining us on this exciting journey exploring valence prediction from fMRI data. We hope you found this research enlightening and informative.